Hello guys, welcome to our channel Mushrooming Your Knowledge. In this video, I am going to discuss about some MCQ type question based upon my previous uploaded videos, that is Prokaryotic DNA Replication Part 1 and Part 2. The link of those videos will be provided in the description box, or you guys can click on the I button. Before I start my question answer session, there is an important thing, that is self-loyalty. After each question, pause the video and choose the option by yourself and then check the answer. So let's start. The first question is, the DNA poly is a template directed enzyme that synthesizes new complementary strand from a parent strand, but it requires the existing short nucleotide sequence for its elongation. Which of the following is required for the synthesis of the primer? Here's the option. The first one is primase or DNA polymerase. The second one is DNA synthase. The second one is RNA synthase, the third one is DNA synthase, and the last one is helicase. So now pause the video and choose your desired one. The correct option is answer A, that is the option A, that is primase or RNA polymerase. Here's the second question. DNA pol3 holoenzyme possesses, here's the option, polymerase activity only, B 3' 5' endonuclease activity, C 3' 5' exonuclease activity and polymerase activities, and the fourth option is 5' 3' exonuclease activity, and the correct answer is option C, that is 3' 5' exonuclease activity and polymerase activities. The third question is, in prokaryotes, the RNA primer from the lagging strand is removed and replaced by the DNA sequence. This process is catalyzed by an enzyme dash, which possesses 5' to 3' exonuclease activity and 5' to 3' polymerase activity. Here's the option. The first one is DNA pol 1, second one is DNA pol 2, third one is DNA pol 3, and the fourth one is DNA pol 4. So the correct answer is option A, that is DNA pol 1. So go for the fourth question, that is, as the two strands of the double helix are separated, the positive supercoiling interferes with the further unwinding of DNA. Which of the following enzyme makes a break in a strand of DNA to release the supercoiling and facilitate the replication to occur? The options are DNA A protein, B SSB proteins, that is single stranded binding protein, the third one is DNA pol, and the fourth one is topoisomerase. So the correct answer is option D, that is topoisomerase. The fifth question is, the short strand of dash primer is required for the replication of DNA. The options are A DNA, B RNA, C histone, and the fourth one is hnRNA. The correct answer is answer B, that is the option B, RNA. The sixth question is, in prokaryotes, DNA replication begins at a single site that is rich in AT nucleotide sequence, where two strands unwind and separate. This ATP dependent process catalyzed by a protein. The options are A DNA protein, B SSB protein, C DNA pol, and the fourth one is topoisomerase. The correct answer is the correct answer is option A, that is DNA A protein. So go for the seventh one. Which of the followings are characteristic features of DNA replication? The options are A DNA replication is template directed, B DNA replication requires short RNA primers, C DNA replication is a highly accurate process, and the fourth one is all of the above. So the correct answer is all of the above. The eighth one is, the DNA replication occurs in a semi-conservative manner, which means the first option is two daughter cells with one consisting of double helical parent DNA, others have newly synthesized DNA. The second one is two daughter cells each consisting one parental strand and one newly synthesized DNA. The third option is two daughter cells each consisting of one half parental and the another half newly synthesized DNA resulting from the crossover. And the fourth option is none of the above. So the correct answer is option B, that is two daughter cells which consisting one parental strand and one newly synthesized DNA. Here's the ninth question. Aminoglycoside antibiotics such as kanamycin, tobramycin, neomycin are known inhibitors of DNA synthesis. It contains a cyclitol ring linked to a 5 or 6 membered sugar by glycosidic bond. These antibiotics are, the options are positively charged compound that to DNA and intercalates it. The second option is positively charged compound that bind to DNA polymerase activity. The third option is negatively charged compound to DNA and denatures it. And the fourth option is negatively charged compound that binds to histones. Here's the option, that is the option A, positively charged compound that to DNA and intercalates it. The 10th question is, in some viruses, RNA serves as the storage of genetic material and DNA is synthesized from RNA by the enzyme known as A. DNA synthetase, B. DNA polymerase, C. reverse transcriptase, and the D is DNA convertase. So the correct answer is answer C, or option C, that is the reverse transcriptase. The 11th question is, a single strand nick in the parental DNA helix just ahead of a replication fork caused the replication forks to break. Recovery from these calamities requires, here's the option, A. DNA ligase, B. site-specific recombination, C. DNA primase, and D. homologous recombination. The correct option is answer D, that is the homologous recombination. The 12th question is, in semi-conservative mode of DNA replication, two parental strands unwind and are used for synthesis of new strands following the rule of complementary base pairing. Synthesis of complementary strands require that DNA synthesis proceeds in opposite direction while the double helix is progressively unwinding and replicating in only one direction. One of the DNA strand is continuously synthesized in the same direction as the advancing replication fork and is called leading strand, whereas the other strand is synthesized discontinuously in segments and is referred to as lagging strand. These short fragments made discontinuously are labeled as Okazaki fragments. These Okazaki fragments need to be matured into continuous DNA strand by which of the following combinations of enzymes? Here's the option, DNA pol 3 and DNA ligase. The second one is DNA pol 2 and DNA ligase. The third one is DNA pol 1 and DNA ligase. And the fourth one is DNA guidase and DNA ligase. So the correct option is option C, that is DNA pol 1 and DNA ligase. The 13th question is, in E. coli, REC-A gene is involved in recombination as well as repair, and DNA B gene is involved in unwinding of DNA double strand during replication. Which of the following statement is or are correct about REC-A and DNA B? The statements are P. Mutations in E. coli, REC-A gene is lethal. The Q statement is E. coli, which mutated DNA gene does not survive. The R is DNA B after uncoiling DNA double strands prevents further reannealing at the separated strand, and the S is REC-A gene is involved in SOS response and helps DNA repair. So the options are, first one is Q and R, second one is Q and S, the third one is P and Q, and the fourth one is P and R. So the correct option is B, that is Q and S. The 14th question is, during DNA replication, events at the replication forks require different types of enzymes having specialized functions, except the options are DNA pol 3, B DNA ligase, C DNA guidase, and the fourth one is DNA glycosylase. The correct option is Option D, that is DNA glycosylase. The 15th question is, on the opposite side of the E. coli chromosome of Odyssey is a dash of termination sequence. The first option is pair, the second option is tertiary structure, third one is single structure, and the fourth one is quaternary structure. So the correct option is option A, that is the pair. The 16th question is, origin of replication usually contains A. GC rich sequence, B. both AT and GC rich sequence, C. no particular stage of sequence, and the fourth one is AT rich sequences. The correct option is D, that is AT rich sequences. The 17th question is, sigma subunit of E. coli RNA pol does not. The options are, first one is initiate transcription and fall off during elongation. The second one, increase affinity of the core enzyme to the promoter. The option C is bind to DNA independent of the core enzyme, and the fourth option is ensure specificity of transcription by interacting with the core enzyme. The correct option is answer C, that is the bind to DNA independent of the core enzyme. The 18th question is, in E. coli grown under nutrient-rich conditions, replication of entire genome takes about 40 minutes, yet it can divide it every 20 minutes. This is so because the options are A. While E. coli divides every 20 minutes, equal transfer of genetic material occurs only in the alternate rounds of cell division. Option B. A second round of genome replication begins before the completion of first round of replication, and by the time cell is ready to divide, two copies of the genome are available. The third option is genome replication and cell division are not coordinate with each other, and the fourth option is during cell division only one of the strands of the genome whose synthesis can be achieved in 20 minutes is transferred to the daughter cell. So the correct option is option B, that is a second round of genome replication begins before the completion of first round of replication, and by the time cell is ready to divide, two copies of the genome are available. Go for the 19th question, that is the prokaryotes have histone-like proteins, that is HV, HH, HU93 or HU90. So the correct option is option C, that is HU93. And the last question, that is the 20th question, the semi-conservative nature of DNA replication was established by Michelson and Stoll in their classic experiment with bacteria. They grew bacteria in N15NH4Cl containing medium, washed and then incubated in fresh medium with N14 containing compounds and allowed to grow for 3 generations. CSCl, that is cesium chloride, density gradient centrifugation of isolated DNA established the nature of semi-conservative DNA replication. The pictorial representation below shows the position of differently labeled DNA in CSCl density gradient. That is, the first one is N14 DNA, second one is N14 by N15 DNA, and the third one is N15 DNA. Had the DNA replication been conservative, what would have been the pattern? Here's the options. The first one, A option, B option, C option, and D option. So the correct option is answer C. So thank you guys for watching our video. If you guys like our content, so then please click on the like button and hit the subscribe button and press the bell icon. Thank you.